The devices, as we prepared them, are packaged into kits of 10 or 20. When they arrive, they're shipped cold in a box. And the kit contains all the components that you need to run the experiment. In the kit is a packet with all the syringes and buffers which can remain at room temperature. There's also a copy of the protocol that we're going to follow in this experiment. There's also a refrigerated box which contains the actual devices and all the running buffers which need to be refrigerated. Typically, kits are packaged in groups of 10 or 20. When not in use, the kits should be refrigerated. Each syringe and each kit component is carefully labeled, and the labels correspond to the table in the protocol that we follow. So there's no ambiguity as to which syringe or kit component should be used in the individual steps. After unpacking the devices and placing them into a biological safety cabinet, we'll be performing the procedure of isolating the neutrophils and lysing them for downstream genomic applications. Because the lysate that we get will be used for isolating, or we will extract the RNA from the cell lysate.